Okay, so the lipid part I'm talking about, the phospholipid that I'm talking about, all right, this is the head portion, and these are the two fatty acids. Now the head portion, it likes water. Okay, so we call it as hydrophilic portion. Hydrophilic. Okay, so if you look at the arrangement here, the way they are arranged, the head portion here, can you see that? The head portion, like, this is cytoplasm. So cytoplasm, of course, will have water, correct? So if you look at the head portion is towards the water part. It likes, it is hydrophobic, hydrophilic. Hydrophilic means water loving. Look at the fatty acids. They are arranged in between here, correct? So the fatty acids, basically, these are hydrophobic. That means they don't like water. They repel. They are hydrophobic. That's why they get arranged, in this particular form, inwardly directed. Okay, so this is all about the phospholipids, and you have proteins which are attached here, all these proteins. And I told you the role of the proteins. The role of the protein is, when it comes to any membrane, role of the proteins will be transporting ions or glucose or any molecule, be it, chloride ion, phosphate ion, enzymes, okay? They all are transported via these protein molecules. So proteins act as transporters, ion channels. Proteins have many roles to play. They're also enzymes in nature. Okay. Some of the proteins also act as receptors. They act as receptors. Receptors of some hormones and all. Okay. So they also act as receptors, transport channels, enzymes. All right. With this, let's come to mesosomes now. Mesosomes, I already told you, the inner membrane is showing you folds, correct? Folds of the inner membrane is mesosome. Respiratory centers, I already told you. Respiratory region. That means it helps in ATP formation because bacteria does not have mitochondria. Now, it is just mesosomes which are involved in, cell wall formation. They help in cell wall formation in the new bacteria. They help in cell wall formation during cell division. So during cell division, a new bacterial cell will be formed, correct? So for that, new cell wall has to be arranged. Mesosomes do this work, okay? So mesosomes are nothing but, I told you, infolds of the inner membrane, of the inner plasma membrane. Okay, they help in respiration, as I told you. They increase the surface area. Now, photosynthetic bacteria, photosynthetic bacteria, these basically have something called as chromatophores. Chromatophores. Now, what are these chromatophores? They are also membrane extensions only. They are also plasma membrane extension only. Okay. But photosynthetic bacteria, I told you, example of a photosynthetic bacteria is cyanobacteria. They can basically make their own food. They have these chromatophores in them. So chromatophores are nothing but they contain pigments in them, and they can help in these chromatophores help in photosynthesis. Okay. So chromatophores basically have pigments on them which help in photosynthesis. And the photosynthetic pigments are nothing but bacterial chlorophylls. They're called as bacterial chlorophylls. You have talked about a photosynthetic bacteria. You get that? Like a cyanobacteria and all. So basically what they have is bacterial chlorophylls. That is a photosynthetic pigment, bacterial chlorophylls. All right with this? Let's come to the flagella. Have you all finished?
Yeah, ma'am.
Okay. So as I said, the entire flagella of a bacteria, which is based on which we say that the bacteria is motile or non-motile, okay, health and movement, they have three parts: filament, hook, and the body. Body is this down. So body is embedded in the outer membrane, peptoglycan, okay, and the plasma membranes. The body is embedded. They're made up of rods and rings. Hook, into the hook is attached the filament, okay. Now, based on how many flagella does a bacteria have, we have a classification. So look at this. First is Ertrichus. Ertrichus means no flagella. Monotrichus, one flagellum at the end. Can you see? Ertrichus, monotrichus, one. Mono means single. Amphitrichus, flagella at two ends. Amphi. Lophotrichus, you have a tuft of flagella. Tuft means many at one end. Tuft. Lopho. So usually when it comes to LOFO, okay, we use this LOFO, no, triggers when they're present at both ends. Okay, this is a little wrong here, this picture. They're present as at both ends. And this is present only at one end. So cephalo means they're present only, a tuft is present only at one end. Lophotrichus is when they have tuft at both ends. Okay, so just, or else you can change, cephalo to lopho and lopho to cephalo. You can draw this out. Okay. So cephalo is when you have a tuft which is present only at one end. Lopho is when you have tuft at both ends. All right. Peri is when you have single all over. Peri means all over. Flagella which is present all over the surface. All right. Next, your work will be, okay, to read about differences between plant and an animal cell, which I'm sure from the very beginning, from 5th standard, you all are reading that, okay? So that is what you should be reading, differences between plant and animal cell. So next class, what I'm going to do is I'm going to start with the plasma membrane structure, okay? The semi-quasi fluid nature of lipids. And a plant cell also. Before that, you have to know the features of a plant cell, okay? And then I'll start with the plant cell wall, animal cell wall. Is that okay? And then we'll start with the
Yeah.
organelles. We'll start with each of these organelles, Golgi and all. All right?
Ma'am, could you scroll to the previous part?
Yes, yes. This?
Yes, yes.
Okay. All right. So the ones who have finished can leave, okay?